This is Lecture 1 for the Spring 2015 seminar, Modern Human Genetics. Today I will introduce three important terms: genotype, phenotype, environment, and how they relate to one another. These are terms you encountered in your introductory genetics courses. The fundamental law of genetics is that phenotype equals genotype plus environment, and I will explain this concept in this lecture. Genetics is the study of biologically inherited traits. A trait is some characteristic of an individual organism, and all living organisms resemble their parents, inheriting some of their traits from their mom and dad. How does this inheritance work? How do we explain the similarities and differences between individuals? Most importantly, genetics is a science. The fundamental unit of genetics is the gene. A gene typically encodes for a protein. Genes come in different forms or variants called alleles. The term allele is important to understand. To make an analogy, a car consists of many parts, which form the whole car. Each part comes in different types. For example, there are different types of tires. The term allele is analogous to type of part. The genome consists of all the genes in an organism along with information how the genes are regulated. Human genome contains approximately 20,000 genes. Genes come in different variants or alleles because they can be altered by mutation. A mutation is a heritable change in a gene caused by a change in the DNA sequence. This change in DNA sequence can change the protein sequence, function, or expression. The term wild type refers to the most common gene allele. Typically, it is thought of as the non-disease gene form, or normal. A mutant gene contains one or more mutations that change the wild-type gene sequence. The three central terms of today's lecture are genotype, phenotype, and environment. Genotype refers to what gene variants or alleles an individual possesses. My genotype is different from your genotype because for some genes I possess different alleles from you. For many, if not most genes, we possess the same alleles. For example, I possess two copies of the A allele for the blood type gene, and my blood type is A. Some of you may possess two copies of the O allele, and your blood type is O's. Others may be homozygous A like me. Phenotype is the observed trait or traits. Environment is the world we live in, which can influence our traits. The goal of genetics is to understand the relationship among genotype, phenotype, and environment. The fundamental law of genetics is that the phenotype equals genotype plus environment. Or more precisely, an individual's traits can be explained by the contributions of genetic factors, or what gene alleles an individual possesses, plus the contribution of environmental factors, for example, what you might eat. For some traits, the environmental contribution is negligible, and so the trait is primarily determined by genetic factors. Can you give an example of this? For other traits, it is the genetic contribution that is negligible, and so the trait is primarily determined by environmental factors. Can you give an example of this? Most traits are determined by a combination of genetic and environmental factors. We will now go over an example of how genotype environment can influence phenotype. The CCR5 gene encodes for a chemokine receptor that is found on CD4 T cells. It is not an essential gene, however, and people can live with a mutant version that does not make the receptor. The AIDS virus, HIV, uses the CCR5 receptor to enter the T cell, resulting in an infection. The main two alleles of the CCR5 gene are the wild-type allele, denoted by the plus, which makes receptor, and the delta 32 allele, denoted by the D32, which contains a 32 base pair deletion and does not make receptor. People who contain two copies of the delta 32 allele, and remember humans are diploids who contain two copies of every gene except for those on the X or Y chromosome, people who contain two copies of the delta 32 allele cannot be infected by HIV, no matter how much they are exposed to the virus. For this example, the phenotype in question is HIV positive or HIV negative. In other words, infected with HIV or not infected with HIV. What is the relationship among genotype, environment, and phenotype for this trait? Assignment 1, which is to be turned in for class on Tuesday, is to answer this question. First, open a Google document. Save it as Assignment 1, MCDB 194HG, and then share it with me. My email address is taumui@gmail.com. Explain the phenotype of HIV positive or HIV negative in terms of an individual's CCR5 genotype and environmental factors. Is this trait primarily determined by genotype? Is it primarily determined by phenotype? Or is it determined by both? Please explain. To help you with this question, you might want to consider the following. For an individual with 2 wild-type copies of the CCR5 gene, what are the possible phenotypes? Does environment matter? For an individual with 2 mutant copies of the CCR5 gene, what are the possible phenotypes? Does environment matter in this case? Please turn in your answers before class on Tuesday. To summarize Lecture 1, I have defined the terms gene, genome, allele, genotype, phenotype, environment, wild type, and mutant. These are terms you should already be familiar with from your previous genetics classes. These are terms that you need to be fluent with for this class. The fundamental law of genetics is that an individual's phenotype or traits can be explained by the contributions of genetic factors or genotype plus environmental factors.